Hi, my name is Carolyn Valek, and I am the support group facilitator for the Worthington, Ohio Myotonic group. And I welcome all the Ohio members on the call. But we also opened up the call to anyone in a support group throughout the U.S. So welcome to you as well. I'd like to introduce our distinguished speaker today, who's on the line and will be doing a presentation. His name is Dr. W. David Arnold, and he's a neurologist, among other things, at Ohio State University Medical Center. Dr. Arnold completed his undergraduate degree at the University of the Cumberlands, where he majored in biology, minored in English, and also competed on cross-country, track, and swimming teams. He completed his medical degree at the University of Louisville, and his physical medicine and rehabilitation residency training at the University of Louisville and Fraser Rehabilitation Institute. He continued with subspecialty training in neuromuscular medicine at Ohio State University, and thereafter joined the faculty. Dr. Arnold has faculty appointments in the departments of neurology, physical medicine and rehabilitation, physiology, and cell biology, and neuroscience. So he's quite a Renaissance man. He's going to be talking today about his research that he does at Ohio State with myotonic dystrophy. Dr. Arnold, welcome.
Thanks for the introduction. It's a great pleasure to be here under the circumstances and in the format. So thanks for everybody for joining. I appreciate it. So I am currently an associate professor at the Ohio State University. I am in several different departments. I primarily work in the Department of Neurology. In fact, I'm actually not a neurologist. I am a physical medicine rehab specialist, but I have kind of lived at OSU in the Department of Neurology. So a lot of times people do think that I'm a neurologist, but in fact, my primary specialty is physical medicine rehab, and then I did subspecialty training in neuromuscular medicine. I've been at OSU for a little over 10 years. I do a combination of clinical work. I see patients every week, but the bulk of my time is spent in research, both clinical research and preclinical research. My clinical research is primarily focused on myotonic dystrophy. In the lab, we study a combination of things, including myotonic dystrophy, as well as what causes muscles to become weaker as we all get older, what we call sarcopenia. So I'm not actually going to talk a whole lot about what I'm directly doing. What I kind of wanted to do is just give a overview of myotonic dystrophy and kind of talk about what causes myotonic dystrophy. Some of this may be a little bit basic for some people. Most people might be quite actually familiar with a lot of things I'm talking about, but hopefully I can add some understanding in regards to what we know and what we don't know, and and kind of give an overview of the progress in the field and some big areas of need. So most people in the audience probably know this, but myotonic dystrophy, myo means muscle, tonic means tone, and dystrophy, of course, means lack of trophic effect or degeneration. And so myotonic dystrophy is named because it's a muscular dystrophy, and there's also increased tone of muscle. And so myotonia, a lot of people have never heard of that. Most people here probably are very familiar with that. But basically, when the nervous system sends a signal to the muscle should respond and one time, but instead of a single response, the muscle continues to contract. And so the tone is increased and the muscles don't relax like they should. There are two main types of myotonic dystrophy, type 1 and type 2. Most people kind of consider three main types because myotonic dystrophy type 1 is split into an early, more severe form, congenital myotonic dystrophy, and then there's later onset forms of myotonic dystrophy type 1. Myotonic dystrophy type 2, it has a lot of overlap in symptoms. Primarily going to focus on myotonic dystrophy type 1 today, because that's the main area of my research. So I think it's always important to highlight this concept. myotonic dystrophy is named a dystrophy, but in fact, it's a lot more than a muscular dystrophy. I would actually say the name is very misleading because some of the effects of myotonic dystrophy are much more prominent, and they have nothing to do with skeletal muscle or the muscles in our arms and legs and so on. That is a major part of the disease, but for some people, the other non-muscle effects are actually even more prominent. So myotonic dystrophy can affect the heart. This is usually not a problem with the pumping action of the heart, although it can be in some cases. And this is normally a problem where the parts of the heart can't communicate like they're supposed to, so the rhythm of the heart gets disrupted. This can be kind of asymptomatic until there's a problem. And so this is something that's really important to have routinely screened to make sure there's not a problem that goes undiagnosed, because this is a very serious problem that can be a significant cause of death if there's a sudden problem with the rhythm of the heart. And it's a very treatable problem, so it's important to screen it because there's an actionable thing that we can address in the clinic. Breathing and pulmonary problems are also a major issue. Not everybody, but as the disease becomes more severe or is more severe, the breathing muscles can be affected more, which can cause inadequate breathing, weak cough, apnea, or kind of periods of not breathing effectively, particularly at night, is associated with the weakness, but also the signals from the brain sometimes don't go to the breathing muscles like they're supposed to. So you can have what people call central apnea. Of course, cataracts are a major symptom for a lot of people. Sometimes these are asymptomatic. Sometimes they do require treatment. And sometimes these symptoms of cataracts are one of the first symptoms that people are kind of trigger the diagnosis. Gastrointestinal problems can be a wide range of problems, including weakness of the muscles of swallowing, but also symptoms that are becoming more and more recognized. And for some people, these are one of the biggest problems. And these can be, IBS symptoms, so like an irritable bowel-like syndrome. Gallstones are more common, constipation and abdominal pain, even to the extreme where contents don't move through the gastrointestinal tract and can almost cause like a small bowel obstruction can occasionally happen. And why it happens is whether it's affecting the muscles of the intestines, which help move the contents, or whether it's related to a neurologic impairment of the movement of the intestines is unclear, and really is a major need to understand that better. The endocrine system can be affected. You can have problems with abnormal thyroid function, poor insulin sensitivity, similar to like a mild form of kind of diabetes. The central nervous system is actually one of the areas that people have really developed more emphasis over the last couple of years, because a major symptom that a lot of people have that sometimes people don't realize is attributed to myotonic dystrophy is the symptoms of fatigue. And that can be really, really problematic in day-to-day activity if you're fatigued with regular activities. So myotonic dystrophy is a genetic problem, so it's hereditary. It's related to a dysfunctional copy of the DMPK gene. We're going to talk about how that dysfunction occurs and what problems it triggers. This is inherited from one parent. And it is a problem where you can see the picture on the screen here. If I can get my pointer to show up. I don't know if it's going to. Well, if you look at the picture on the screen, you can see the DMPK. That's basically in orange is the gene that is mutated in myotonic dystrophy. And in one part of that gene, there's a sequence of nucleotides. And nucleotides are basically the reading part of DNA. So the kind of the what makes up the message of DNA. And in one region of that gene, there is a expanded CTG repeat, so a repeat expansion, and it can be anywhere from a small number of repeats to a very large number of repeats. And that has a functional consequence. So if you look at the number of repeats in the DMPK gene, the gene that is mutated in myotonic dystrophy. If the number of repeats are 37 or less, then there's no diagnosis of myotonic dystrophy type 1. If it's in the middle between 37 and 49, individuals with those repeat size don't have symptoms, but they're at risk of passing it on to future generations. At a count of 50 or greater, that's when the diagnosis of myotonic dystrophy is confirmed. And as you get more and more repeats, the symptom severity and the onset of the disease in general is earlier and more severe. So how do the CTG repeats cause myotonic dystrophy? So I'm going to try to keep this as simple as possible, but as Einstein says, I don't want to make it too simple, because if you oversimplify, it can be inaccurate. So what I want to try to do, and I'm planning on leaving plenty of time at the end for questions if there are things that are not clear, so that we can clarify things. That hopefully, that doesn't happen. But if it is not clear, then we can hopefully clarify things at the end. So what I'm going to do is kind of step back and give just a little bit of overview of genetics. For some people, again, this is probably going to be a boring review, hopefully not too boring, but it's important to make sure everybody's kind of on the same page before I talk about what we know and what we don't know about myotonic dystrophy. So genes and DNA, genes are basically the instruction for how cells know how to make different proteins or do different biological processes within that cell. So in each cell, there are approximately 20 to 25,000 genes. Many of these genes make proteins. Many of these genes do not make proteins. So not every gene is associated with a protein product per se. Each cell has 2 copies of each gene, one from mom and one from dad. If there is one dysfunctional gene that's related to the mutation with myotonic dystrophy, then that causes the disease. And when that happens, when only one copy is required to cause a disease, we call that autosomal dominant. It's not really that important to remember that term, but most people will hear that myotonic dystrophy is autosomal dominant disorder. There are autosomal recessive disorders and other types of inheritance patterns. And what that just basically means is you have those 2 copies. If one's not working like it's supposed to, it kind of causes a problem that kind of overrides the other copy, and then it results in whatever autosomal dominant disease you're talking about. So this is a really simple illustration to kind of highlight what a gene does in the body, and try not to make this overly simple, but also this is going to set us up to talk about what happens in myotonic dystrophy. So basically, a gene is a recipe for making something or doing something in the cell. So a cellular process or making a protein. And each cell actually has the whole cookbook of all the genes in the body. But that cell will only use the genes that it needs to use. And each gene is made up of DNA, which is basically the kind of the words in the cookbook, which are made up of nucleotides, which is kind of like the letters of the alphabet. And so when the nucleotides are mixed up, then the words get mixed up and things don't work like they're supposed to. And in some genetic problems, that's actually what causes the dysfunction. But in myotonic dystrophy, it's actually a little bit different, because instead of being mixed up, there's extra nucleotides, extra words. They're kind of in the middle of the recipe. One other comment that I would point out here is each gene has one function, makes a protein, but actually that function of that protein can be a little bit different. And so I kind of liken it to making cookies. There's different types of cookies. So we're going to kind of use that analogy to describe how DNA goes to making a protein. So you have this cookie gene, and I apologize, I don't have a pointer, so that makes it a little bit more difficult. I don't know why I don't have that, but basically on the top row, we have the DNA. And so the DNA for every gene is in every cell. So this is what we're going to call the cookie gene, a simple illustration. And you can see the different colors. So the different colors are the different exons of a gene. So different genes have different numbers of exons. Basically, that's the part of the gene that actually gets directly turned into part of the protein. So the body takes the DNA and kind of makes a copy of it. And you can see that there's the arrow that says from the DNA to the pre-mRNA, it says transcription. So transcription is just that. It's making a copy of the DNA into RNA. And the first copy actually has way too much information in there for the body to be able to use it. And that's the reason it's called pre-mRNA. mRNA stands for messenger RNA. And so that big copy with all the extra information in there undergoes what's called splicing, and basically takes out the extra parts that are not needed in this cell or that cell, wherever the gene is being turned into a protein. And so you can see down below, I have 3 different proteins here, and these are all coming from the same gene, and depending on which cell is expressing this gene, it may turn to different types of protein, or in this case, a different type of cookie. So in one cell, you may need a chocolate chip cookie. One person might want chocolate chip. Another person might want peanut butter. But it's very important that the person that is getting that cookie gets the one that they want. It's very important that the gene is expressed the way it should be in a particular cell or another cell. And over to the right, you can see the purple kind of oblong shapes. That's representing an RNA binding protein. What that basically does is it helps kind of cut this big long copy of the DNA, the pre-messenger RNA, pre-mRNA, and shortens it and makes it into the right version. So to make sure that you're doing the right ingredients and making the right type of cookie. So what causes DM1? So now that was an overview. We'll go back through that again as we explain what happens in myotonic dystrophy. So in this DMPK gene, there's extra CTG repeats. That's the different letters of the DNA alphabet that make up the words in the cookbook. And so there's extra letters in there that really aren't in the main part of the recipe. They're kind of off to the side. And so it's not like it's breaking up the actual recipe. It's doing some other things, which we'll talk about. So again, here's an example, that same picture I was showing you before. But I'm going to kind of start to talk about one of the main mechanisms of what we think is happening in myotonic dystrophy. So again, on the top line, we have DNA. That is the whole copy of the gene that's in each cell. And that cell makes a copy of it into RNA. And then that big copy is trimmed down into individual copies. So in myotonic dystrophy, these extra repeats get kind of copied. And then, so between the DNA step and the pre-messenger RNA step, they get copied into these structures that are down in your bottom left-hand corner. So the blue kind of hairpin color, we call it RNA repeat hairpin structure, and they kind of fold against each other. And then they form these little pink dots that are in kind of the middle panel down at the bottom. So this is a picture of cells and showing these what we call RNA foci, or basically globs of these RNA repeats. And then they bind and they kind of clump up the RNA binding proteins. So when you're trying to take your copy of the DNA, the pre-messenger RNA, to the mature RNA where it's made exactly how it's supposed to make the right type of cookie, the RNA binding proteins are very important for that. And so when they're not present, then the wrong kind of cookie can be made in the type as compared to the type that needs to be made in whatever cell that's expressing this gene. And so these repeats, the CTG repeats, form these kind of foci that kind of grab up all the RNA binding proteins, because you can see on the picture down in the bottom left, the blue is the RNA, the CTG repeats, and they're sticking together and making that hairpin structure. And then the pink around the edge is the RNA binding proteins that are needed for this splicing, but because they're stuck there, they can't do their job. So what happens to the cookie gene recipe in myotonic dystrophy? So I had my son here help us with this. So this is a picture of Liam down the right-hand corner. So these are 2 copies of the recipe that I showed you before for the cookie. So what can happen is basically part of the recipe that's needed to make a certain type of cookie can be kind of cut short, or doesn't have the parts that that are needed. And so you can see here, Liam didn't really have the patience to write the whole recipe. And so the gene, the copy of the RNA wasn't trimmed correctly, so the splicing was abnormal. What we call that is missplicing. So it's missplicing for the type of protein that cell needs. So this is actually one of the main things that we think contributes to the multi-system effects of myotonic dystrophy. So on this slide, you have the very nice illustration of the different effects that myotonic dystrophy can cause from the Myotonic website. And you have the blocking of the RNA proteins on the top right-hand corner. And then on the bottom, don't get too caught up into the details, but basically that MBNL1, that's what we call muscle blind. And then you have CUG-BP1. Those are two RNA binding proteins that are normally kind of in balance. But when a cell is affected with myotonic dystrophy, the muscle blind levels get too low, and then the types of proteins that are made for different genes are shifted to one side more than they should be. So basically the types of cookies that are made for different cells, or the types of proteins that are made by these genes in different cells, are not the ones that need to be made. And so you get this kind of misregulation of what types of proteins are being made. And so there's several different genes that are affected, and that's at least part of the reason why different parts of the body are affected in different ways. And so some of these are, if you look down at the bottom, the CLC-1, that's the chloride ion channel that is in muscle. And when you have that change from the left to the right, you have that kind of black rectangle in the middle, it basically blocks the production of that channel, which in a muscle. And so what that channel basically does is helps muscles relax. And so when it's not there, that's when you get the myotonia, that's when you get the stiffness of muscle. And this is really the best described mechanism of what's happening in myotonic dystrophy. It's these RNA binding proteins are getting disrupted, so splicing is changed, and so different genes are not working the way they're supposed to, not being expressed the way they should be. I'm not going to go into some other mechanisms that are also probably at least partially contributing, some maybe more than others. There's a newer concept called RAND translation. Some people probably have heard of that. That's basically what happens with this is the CTG repeats in the DMPK gene, normally they're not turned into a protein. They're in part of the gene that's just normally cut out. But because of this repeat, it sometimes tricks the body into making proteins from those repeats. And there's pretty good evidence that in myotonic dystrophy 1, 2, and other diseases too, that this RAND translation actually produces these toxic proteins that are probably causing deficits in cells as well. So that's also another contributing factor to myotonic dystrophy likely. There is a little bit of evidence. Also, the DMPK gene, if you lose function of that gene, which is where these repeats are, that could also cause some problems. But that probably doesn't explain most of the problems in myotonic dystrophy. There are some other genes that are also close to that DMPK gene, and these repeats might also inactivate those genes as well. And so that's also been considered a possible mechanism. So now we kind of know at least part of what causes myotonic dystrophy and the multi-system effects. How could we potentially fix that? So the easiest, well, let's not say the easiest. Let's say the seemingly easiest would to be what people now call genome editing. So basically take out the CTG repeats using something like CRISPR-Cas9. And so then you don't have these RNA foci in the cells that are blocking the splicing machinery of the cells. Another thing that we haven't talked about yet, but these repeats get longer between generations. So from a parent to a child, generally they get longer. They also get longer within a person across the lifespan. They also get longer in different tissues at different rates. So this CTG repeat expansion is unstable. So it actually gets longer within the same person. So if we could do something to stabilize the repeat length, that's one thing that could potentially reduce the progression of the disease. The second possibility, so first reduce the number of repeats, kind of just cut them out, edit it. The second is to stabilize the number of repeats. And those two problems are really pretty far off because genome editing, the challenges with that is you don't want to have off-target effects. So if you are editing the different genes, if you have an off-target editing, so it's not editing the right thing, or you have it corrected incorrectly, then that can be really problematic. So that's far off, but still, if it gets optimized, might be something in the future that could be really exciting. The next strategy is decreasing the RNA foci. So that could be accomplished by several different things. So when we make this copy of the DNA to the immature pre-messenger RNA, you could prevent the extra repeats, the CTG repeats, from being transcribed or prevent transcription. Another thing is down at the bottom, you can see kind of the little red dots. That's the muscleblind, those RNA binding proteins. You can kind of prevent the interaction of that CTG repeat RNA and the RNA binding protein. So kind of free up the muscleblind RNA binding proteins that are being kind of sucked up into these foci or sequestered into these foci. And then the next one is breaking up the RNA repeats, and that is actually the one that a few years ago in the Ionis trial, where they used the antisense oligonucleotide, it was targeting these repeats and trying to get the body's machinery to break up those repeats. And in fact, it was a very nice strategy. The biggest challenge was the antisense oligonucleotide, which is kind of like it kind of is a copy of the repeat, and it kind of sticks to the repeat. The problem with it did that very well, but it didn't get to the tissues that it needed to do its job. So it was very good at doing what it did. But when we did muscle biopsies of individuals in the trial, we could see that the antisense oligonucleotides didn't get to the muscle like they needed to be, and probably didn't get to other tissues, but we can't biopsy other tissues as easily as muscle. And so one of the big areas right now is trying to figure out how to get better distribution. So basically, if you deliver this antisense oligonucleotide therapy or some other therapy, it has to get to the tissue. So optimizing it so the distribution is better. Another one that's theoretically might make sense, might not make sense, but these binding proteins, these RNA binding proteins, are getting sequestered or kind of sucked up into these RNA foci. So you could increase the levels of those proteins to kind of overcome that sequestering or that binding interaction. And so there are ways to overexpress proteins in other diseases like spinal muscular atrophy. I was part of the clinical and preclinical work moving gene therapy forward for SMA. And in that, you're overexpressing a protein and you're delivering it with like a virus that's, doesn't cause disease, but just carries the cargo. So you could potentially overexpress RNA binding proteins. But again, that's probably a little bit more indirect than the other possible strategies. So the big needs that really are in the field right now that I think have, it's remarkable how much we know about this disease and how clear we really have the kind of vision of what we have of the mechanism. The biggest challenge is we really don't have good animal models. So a lot of other diseases that have moved forward more quickly, but even maybe less understood as far as mechanism, they have better animal models. And the animal models right now that we have for myotonic dystrophy either don't have a lot of defects for whatever reason that aren't always clear, or they don't have defects that are important to people that are affected with myotonic dystrophy. So the best model would be, has similar problems that individuals affected with myotonic dystrophy have. So then you can make sure that you can fix the things that are most important. Another big area that is really challenging with myotonic dystrophy is the fact that it has all these different symptoms in different people, different systems that are affected, probably because of this missplicing and maybe other factors too, as we've talked about. That makes it difficult to understand the best way to kind of track it when you do a clinical trial. And so we have to develop, as good of an understanding as possible about what's the most important symptoms that affect individuals with myotonic dystrophy, what's the best way to track those symptoms? So then when we have a treatment that might be effective, we can really show that as quickly as possible, because clinical trials are expensive, really demanding for individuals that are involved, and take a long time. So if we have better measures, we can speed that process up. And another important thing is understanding there's a lot of variability between individuals. Some of this could be from this unstable repeat expansion between different people, but there are possibly other factors too. And so there are big natural history studies. In particular, we're involved with the one led by Nick Johnson, unfortunately kind of on hold with all of the things that are going on right now, but really exciting. It's going to be a really big study across the world at multiple different sites to try to understand some of these things, so we are really as prepared as possible as the therapies are tested in preclinical studies as they move forward to the clinic. So you can keep track of this on the Myotonic website. It's also important, I think, to stay linked in with a clinic that's kind of on the front end, because they can give you, that's not possible for everybody, but if it's possible, I think it's nice, because sometimes these studies fill up quickly, and it's just good to be kind of connected to a clinic that can give you the most update information. But this website is really, really helpful if that's not possible. One thing I just wanted to highlight of a study, this is actually just a small little study at OSU by one of my colleagues. I'm helping with it, but it's not really me. So Dr. LaRusso is a assistant professor at OSU. We work closely together. We're working on setting up a multidisciplinary clinic that's primarily focused on myotonic dystrophy, kind of still within the under the umbrella of MDA, but we're trying to set it kind of specifically, because I think there's a lot of needs in the area of myotonic dystrophy that are much more complicated and complex than a lot of other populations in the MDA clinic. So we're trying to specifically do that. But this study is a very simple little study trying to understand the effects of nutrition in myotonic dystrophy. So there's a lot of reasons why nutrition could be problematic. One, because of these missplicing problems that can affect the endocrine system, that can affect metabolism, a lot of different things. But also when there's difficulty with swallowing, that can reduce intake, weight loss, malnutrition, things like that, which if you already have problems with muscle weakness, you don't want to have more weakness contributed from malnutrition. So I just want to highlight this real quick. This is kind of ongoing. It's a small study, but we're trying to look at the effects of nutritional status in individuals with myotonic dystrophy compared to age-matched controls. And so it's a series of blood tests to look at nutritional values, as well as body composition using a DEXA scan, which just basically gives you lean mass and bone mass and fat mass, just a one-time visit. This is for individuals between 18 and 60. And the one thing that the one inclusion criteria that might be difficult for some people is we do want to have controls that are living in the same household, because what we're trying to do is kind of control for dietary habits. So we want to see if not just diet is different, but if the effect of myotonic dystrophy with the same diet is significant. So that's one caveat with the study. So that is all that I had prepared. Hopefully that was helpful and not too confusing. And
thank you, Dr. Arnold. That was really great. Really appreciate it. It was really educational for all of us.
Yes.
Thanks for having me.